Good morning, or actually it's almost afternoon, but I want to go over chapter 7, conception and fetal development. I'm going to go over this pretty closely with the book, so you might come follow along. It starts on page 135. There will be several pictures in this presentation that are referred to from your book, so be sure and look at those as we kind of go through them. All right, so what do we know about conception and fetal development? First of all, in order for conception to happen, several things have to be taking place and working as it's supposed to be. So let me get this video on so you can see it. Okay. So to get started, we're going to go over a little bit. I believe this part is in chapter 8, but we're going to talk a little bit about hormones and what their action is and how they affect conception and fetal development. So you guys all heard of estrogens. Estrogens is a hormone that's very widely spread out throughout the body and used for many things. It's produced by your ovaries. Those of you who have ovaries, females have ovaries and produce estrogen. Later, during pregnancy, estrogen is also produced by the placenta, and this helps the uterus to grow. It maintains the lining that's inside of the uterus in order to maintain a pregnancy. Estrogen helps to increase blood circulation, and it also activates and regulates the production of other hormones. In early pregnancy, estrogen is necessary to promote the growth of the breast. So early on, when the female becomes pregnant, you may hear them complain of breast tenderness, breast soreness, and that's a lot of times one of the first signs of a pregnancy. This is due to the release of estrogen. So estrogen also causes the breast to become a little bigger and a little bit more swollen and tender. Later on, during the pregnancy, it helps promote the development of milk making in order for breastfeeding later on. Estrogen also helps regulate the menstrual cycle. It helps to control the growth of the uterine lining during the first part of the cycle. And if that cycle doesn't end in a pregnancy, that your lining is kind of sloughed off from the uterus. Okay, another hormone, progesterone, we hear of quite a bit as well. Progesterone helps to prepare the body for pregnancy in the event that the egg is released and it is fertilized. If the egg is not fertilized, the corpus luteum breaks down and the production of progesterone will fall and a new menstrual cycle will begin over again. If the egg ends in fertilization, then progesterone stimulates the growth of blood vessels that supply the lining of the endometrium in the uterus, and it stimulates glands in the endometrium to secrete nutrients that nourish the early embryo. Progesterone also helps to prepare the tissue in the lining of the uterus to allow the fertilized egg to implant, and helps to maintain the endometrium throughout the pregnancy to maintain that pregnancy. Okay, another hormone you need to be aware of is FSH, follicle stimulating hormone. This hormone is used to it's produced and released by the cells in the anterior pituitary gland. In women, this hormone helps to stimulate the growth of the ovarian follicles in the ovary, and that's before the release of an oocyte from one follicle at ovulation. It also increases estradiol production. In men, progesterone, FSH, follicle stimulating hormone, acts as the Sertoli cells of the testes and just stimulate sperm production. Okay, one other hormone we need to be aware of for pregnancy-related issues is luteinizing hormone, or LH. This is produced and released by cells in the anterior pituitary gland as well. In men, it stimulates Leydig cells in the testes to help produce testosterone. In women, in week 1 and week 2 of the menstrual cycle, luteinizing hormone is required to stimulate the ovarian follicles in the ovary, and that's to produce the female sex hormone, estradiol. Around day 14 of the cycle, a surge in luteinizing hormone, or LH, levels cause the ovarian follicle to tear and release a mature oocyte from the ovary. From the weeks 3 to 4 of the cycle, the remnants of the ovarian follicle form a corpus luteum. Luteinizing hormone stimulates the corpus luteum to produce progesterone as well. Okay, so there's 2 phases of the ovarian cycle. The follicular phase, which is days 1 through 4. This is when the follicle matures and ovulation occurs. Then you have the luteal phase, which is days 15 to 28. It begins with the ovum, it leaves the follicle, and the corpus luteum develops from that ruptured follicle. Here is a picture. This is also in your book. It's a graph that shows the female reproductive cycle. And you can see in the middle there, the ovarian hormones. When those are released at the beginning of the cycle, days 1 to the middle of the cycle, estrogen is increasing and plays a role strongly at this cycle. And then towards the middle to later end of the cycle, progesterone increases and has a big part in the cycle at that time. So just kind of remember that estrogen helps get a pregnancy and the uterus ready for pregnancy, helps get that going early on in the cycle. Progesterone helps to maintain a pregnancy if it occurs. Okay, and this is a very similar graph that shows you the 4 phases of the menstrual cycle. The menstrual, the proliferative, secretory, and the ischemic phase. So menstruation is initiated each month by falling levels of estrogen and progesterone and the release of prostaglandins, which constrict the spiral arteries. This causes them to spasm, those arteries to spasm and contract and break up. The blood supply to the endometrium is cut off, and the cells of the top layer of the endometrium become deprived of oxygen, so they die. That layer is lost or kind of slept off from the uterus. The proliferative phase is the second phase of the uterine cycle, when estrogen causes the lining of the uterus to grow and proliferate. The secretory phase, it occurs from day 14 to about day 28 of the cycle. Progesterone, stimulated by the luteinizing hormone, or LH, is the dominant hormone during this phase and helps prepare the corpus luteum and the endometrium for possible fertilized ovum implantation. Okay, so this is the proliferative phase. Again, endometrial cells enlarge and they become twisted and longer due to estrogen. The endometrium thickens 6 to 8-fold, so it's quite a bit thicker. The cervical mucosa at this time becomes thin, it's clear and watery, and it's a little more towards the alkaline side. In the secretory phase, this follows ovulation, so ovulation has just occurred. You'll see progesterone cause a marked increase in swelling, and the amount of tissue glycogen increases. Vascularity of the uterus is greatly increased during this stage, and And that's due to providing a nourishing bed for implantation. So it's much thicker, more nourishing if that fertilized egg, or if the egg is fertilized, and allows it to implant into the bed of the uterus. The ischemic phase, this is when if implantation does occur, or sorry, if it does not occur, the corpus luteum will begin to degenerate. Estrogen and progesterone levels will start falling. This phase is characterized by the escape of blood into the stromal cells of the uterus. Okay, so moving forward after covering these hormones, you kind of have a reference of what's affecting what during the conception and development stage. So chapter 7. All right, so tetragenesis or tetragens, you've probably heard of those before, maybe called something different like poisons. Tetragenesis is in the embryo is most vulnerable during the first 8 weeks of growth. That's when cell differentiation is occurring. And that's also when the organs are beginning to develop in the embryo. So definitely a problem during that first 8 weeks. The effects of teratogens, such as drugs, different viruses, maybe radiation exposure, those all depend on the maternal and fetal genotype, the stage in development when that exposure happens to occur, and the amount or dose and duration of exposure to a particular agent. So factors that may affect embryonic development include the quality of the sperm or the ovum from which the zygote is formed. The genetic code established at fertilization is also can be affected, and the adequacy of the intrauterine environment. If the environment is unsuitable before cellular differentiation occurs, all the cells of the zygote are affected. The cells may die, which causes spontaneous abortion, or growth may be slowed down. And that's depending on the severity of the situation at the time. When differentiation is complete and the fetal membranes have already formed, an injurious agent has the greatest effect on those cells that are undergoing the most rapid growth. So the time of injury is critical in the development of anomalies. Okay, so preconception counseling is very important for some people more than others even, but everybody needs to be aware of what may harm an embryo or a fetus at certain times during the pregnancy. Modifiable risk factors, there's several that you can control or change in order to decrease the risk to the embryo or fetus. So here are some modifiable risks that can be changed or stopped, such as smoking, alcohol, social and street drugs, caffeine, medications, and environmental hazards. Smoking causes vasoconstriction that can interfere with the placental circulation. The infant going through this may suffer negative effects that include intrauterine growth restriction. However, lung problems should be avoided during pregnancy. Anything that can cause lung problems. All right, so talking about cells and how they divide or multiply in order to go from a single little ova into a person. So this is the process of cellular division. One process is mitosis. There's an example at the top of the screen that shows you how the cell divides and what it ends up with after dividing, which is important when you talk about the different types of cellular division. So this being mitosis, we're going to talk about somatic cells is what we start with. The parent cell divides into 2 daughter cells, and they are the exact copy of the parent cell or the original cell. So they're exactly the same. Each daughter cell has its own nucleus, Again, these cells are identical to the parent cell and to each other. So each daughter cell looks like the other daughter cell, and which looks like the parent. Okay, so one cell divides and turns into two identical cells that are identical to the original cell. They contain the same diploid number of chromosomes or genetic material, which is 46 chromosomes. So they all contain 46 chromosomes. The other type of cell division is meiosis. This cell division produces reproductive cells. So remember, your difference in mitosis and meiosis is meiosis produces reproductive cells, and mitosis is somatic cells. Okay, these reproductive cells are called gametes. That is the sperm and the ova. Each cell contains half or 23 of the different genetic material of the parent cell. It's a haploid. So they contain different genetic material, and they contain half of the amount, which is 23 instead of the 46. Meiosis consists of two successive cell divisions. First division is chromosomes will replicate. And then the second division, chromatids of each chromosome separate and move to opposite poles in each of the daughter cells. So meiosis is cellular division that results in the formation of 4 cells. Each cell contains 23 haploid numbers of chromosomes, and daughter cells contain only half the DNA of the normal somatic cells. So after this occurs, we have pairing that takes place. And this takes place between homologous chromosomes. Instead of separating immediately, as in mitosis, the chromosomes become closely intertwined. At each point of contact, there is a physical exchange of genetic material between the chromatids or the arms of the chromosomes. New combinations are provided by the newly formed chromosomes. These combinations account for the wide variation of traits in people, such as eye color, hair color. So here is an example of mitosis versus meiosis. And as you can see on mitosis, identical, you have 46 chromosomes in each cell. With the replication and the division, they end up being exactly the same. With meiosis, they're not identical. Replication occurs and then cell division. Gametes, which are the sex cells, sperm and ova, and end up with 23 random chromosomes in each cell down here. Okay, so these divide, and remember, they share information at the point of contact when they kind of chain around each other. So those points of contact are different in every cell, and you're going to end up with different chromosomes. So gametogenesis is the process by which germ cells or gametes, such as the ovum and sperm, are produced. These cells contain only half the genetic material of a typical body cell. So each gamete must have only the haploid number, which is 23, of chromosomes, so that when the female gamete, or the egg, ovum, and the male gamete, the sperm, when those unite to form the zygote, which is a fertilized ovum, the normal human diploid number of chromosomes, which is 46, come back together. So you have half from the mother and half from the father. When they come together, they reestablish the number of 46. Okay, so oogenesis is the process that produces the female gamete, which is also called the ovum or egg. Ovaries begin to develop early in the fetal life of the female. Ovaries give rise to organelle cells and develop into oocytes. During puberty, mature primary oocytes continues through the first meiotic division in the ovary. So that's when one cell turns into two different cells. Haploid cells are released at ovulation. Remember, the haploid is half, 23 chromosomes. All the ova that the female will produce in her lifetime are present at birth. And meiosis, which is cell replication by division, begins in all oocytes before the female fetus is born, but stops before the first division is complete and remains in this arrested phase until puberty. Excuse me. During puberty, the mature primary oocyte proceeds by oogenesis through the first meiotic division in the Graafian follicle of the ovary. So the first division happens, second one is kind of put on hold till puberty. Okay, the first meiotic division happens when it's still in the ovary. Two cells that are unequal size is produced with the same number of chromosomes. One cell is a secondary oocyte, the other is a minute polar body. Secondary oocyte and the polar body each contain 22 double-structured autosomal chromosomes, and they contain one double-structured sex chromosome. Okay, so 22, and then one more of the sex chromosomes. At the time of ovulation, a second meiotic division begins immediately and proceeds as the oocyte moves down the fallopian tube. Division is again not equal, and the secondary oocyte moves into the metaphase stage of cell division, where its meiotic division is arrested again until later on, and unless the oocyte is fertilized. The second polar body, which is also haploid, forms at the same time. The first polar body has now also divided, producing two additional polar bodies. Thus, at the completion of meiosis, 4 haploid cells have been produced, 3 polar bodies, which eventually disintegrate, and then you have 1 ovum. Production of the male gamete or sperm also happens during puberty. The spermatogonium, I'm saying it wrong, probably spermatogonium, the primordial germ cell. This begins with complete set of genetic material. The diploid number of chromosomes, remember, is 46. The cell replicates before it enters its first meiotic division. The cell is now a primary spermatocyte. So again, we're talking about spermatogenesis now. The spermatogonium, a primordial germ cell. During the first meiotic division, it's going to form 2 cells that are called secondary spermatocytes. Each of these contain 22 double-structured autosomal chromosomes and either double-structured X sex chromosomes or double-structured Y sex chromosomes. During the second meiotic division, these divide to form 4 spermatids, each with haploid numbers of 23 chromosomes. Spermatids undergo a series of changes. They lose most of their cytoplasm, and then they become sperm or spermatozoa. The spermatids undergo a series of changes during which they lose again their cytoplasm and they become spermatozoa. This is in figure 7-1b in your book. The nucleus becomes compacted into the head of the sperm. This is covered by a cap that's called An acrosome. The acrosome in turn is covered by a plasma membrane. There's a long tail that's produced from one of the centrioles on the sperm. Okay, this is figure 7.1, Gabatogenesis. So take a look at that in your book as well. It shows you the ova and the sperm and how that the immature diploid cells end up becoming the mature spermatozoa and ovum. Okay, preparation for fertilization. Remember, the ovum is released into the fallopian tube. The ova is considered fertile for about 12 to 24 hours after ovulation occurs. It's viable for 48 to 72 hours after it's in the vaginal tract. Okay, it's very healthy and highly fertile for the first 24 hours. So know that the ovum does not actually move itself. And remember, it's got to make its way all the way through that fallopian tube. Peristalsis of the tubes caused by estrogen helps to move that ova down the tube. Thinning of the cervical mucus that we talked about just a few minutes ago, which is caused by estrogen, also helps that ova to be able to travel through those fallopian tubes. Fertilization occurs in the ampulla, which is the outer third of the fallopian tube. The male deposits about 200 to 300 million spermatozoa into the vagina in a single ejaculation. Only about 300 to 500 sperm actually reach the ampulla. So sperm again can survive in the female reproductive tract for about 48 to 72 hours. We talked about the ovum having no inherent power of its own to move, that it is helped through estrogen by the thinning of the mucus and also the peristalsis waves that are caused in the fallopian tubes. Fructose is in the semen, and that is secreted by the seminal vesicles, and this allows for the high energy source for the sperm. So with the sperm being as active as it is, it does need energy and gets fructose. The transient time from the cervix into the fallopian tube can be about as short as 30 minutes, or it can be as long as 6 days to make that trip. Prostaglandins that are present in the semen may increase uterine smooth muscle contractions, which helps transport the sperm. The fallopian tubes have a dual ciliary action that facilitates movement of the ovum toward the uterus and movement of the sperm from the uterus toward the ovary. The sperm must undergo 2 processes before fertilization can occur, and that's capacitation and the acrosomial reaction. Capacitation is the removal of the plasma membrane that's overlying the spermatozoa acrosomial area, and it's the loss of the seminal plasma proteins. If the glycoprotein coat is not removed, the sperm will not be able to fertilize the ovum. Capacitation occurs in the female reproductive tract, which is aided by the uterine enzymes, and it's thought to take about 7 hours for the process to occur. Sperm that undergoes capacitation now takes on 3 characteristics: the ability to undergo the acrosomial reaction, the ability to bind to the zone of pellucidea, and the acquisition of hypermotility. The acrosomial reaction follows capacitation, whereby the acrosomes of the sperm surrounding the ovum release their enzymes, and that causes the breakdown of the hyaluronic acid in the ova. so the penetration of the sperm can happen to the ova. For conception, it's best to have intercourse several times around ovulation in order to make sure the sperm at its best, most active time meets the ova at its most healthy and highly fertile time. All right, the actual moment of fertilization. Sperm penetration causes chemical reactions that block more sperm penetration, so only one sperm can enter the ova. Sperm enters the ovum, a chemical signal prompts the secondary oocyte to complete the second meiotic division. Remember, it's been on hold. The true moment of fertilization occurs as the nuclei unite. So the chromosomes pair up to produce diploid zygote. Each nucleus contains a haploid number of chromosomes, which remember was 23, and the union restores the diploid number 46, the union of the ova and the sperm. Zygote contains a new combination of genetic material. The sex of the zygote can be determined at the moment of fertilization. Two chromosomes of 23rd pair, of the 23rd pair, which is the sex chromosomes, they either have a double X or XY combination, which determines the sex of the individual. Females will have two X chromosomes, and males will have an X and a Y chromosome. So each of these chromosomes, certain traits are termed as sex-linked because they are controlled by the genes on the X sex chromosome. So two examples of the sex-linked traits are color blindness and hemophilia. And what that means is certain diseases or traits are linked with either female or male. And as we know, color blindness is passed on through the female chromosomes. All right, this is a picture of an ovum being penetrated or trying to be penetrated by a sperm. Remember, only one of those sperm gets in, and when it does, after they go through a certain process, is able to penetrate the ova, and it causes a chemical reaction that doesn't allow any other sperm to penetrate the ova. Okay, the pre-embryonic development. This is development of the little person. Pre-embryonic stage is the first 14 days of development. This starts at the day the ovum is fertilized, or the day of conception. Cell multiplication, the zygote, goes through rapid mitotic division. This is called cleavage. Blastomeres are divided cells that grow to morula. That's a solid ball of about 12 to 32 cells. It's just a little mass called a blastomere. The morula divides into a solid mass, which is the blastocyst, and it's surrounded by an outer layer of cells called the trophoblast. Implantation occurs usually about 7 to 10 days. Okay, let's see. Cellular multiplication begins as the zygote moves through the fallopian tubes towards the cavity of the uterus. This transport takes about 3 days or more, and it's accomplished mainly by a very weak fluid current in the fallopian tube that results from the beating action of, remember, the ciliated epithelium that lines the tube. The outer layer of the cells that surrounds the cavity and replaces the zona pellucidea is the trophoblast. The trophoblast develops into one of the two embryonic membranes, which is called the chorion. The inner solid mass of the cells, called the blastocyst, develops into a double layer of cells called the embryonic disc. And from this, the embryo and the amnion, which is the other embryonic membrane, will develop. Okay, so about 10 to 14 days in, you'll see cell differentiation is happening, and it's going to lead to the production of the ectoderm, the mesoderm, and the endoderm, from which all the tissues and organ systems will be developed from. Blastocyst differentiates into 3 primary germ layers. That's the ectoderm, mesoderm, and endoderm. Again, all tissues, organs, and systems will develop from these 3 primary germ cell layers. Embryonic membranes form at implantation, which is called nidation, and the chorion and the amnion is produced or formed. So you will hear a lot about the chorion and the amnion in the future to come in our classes, because that is something that fetal membrane carries on throughout pregnancy and can have some issues as well. This is figure 7.4 in your book, and that's showing you the formation of the chorion and the amnion. Okay, so this picture 7.4, formation of the primary germ layers is or implantation of the 7.5-day-old blastocyst, okay, into the fluid-filled space. B is implantation is completed by day 9. An extra embryonic mesoderm is beginning to form a discrete layer beneath the cytophoblast. By day 16, the embryo shows all 3 germ layers. It has a yolk sac and an outpouching of the yolk sac that forms the structural basis of the body stalk, or the umbilical cord. Okay, and then let's remember that progesterone, the hormone progesterone, definitely influences the endometrium, the increase in the thickness and the vascularity of the endometrium in order to prepare for implantation, and also to form nutrition for the ovum. Okay, that's progesterone. All right. Other things that are also produced from these layers: amniotic fluid. The amnion and the chorion grow and connect, and they form that amniotic sac to produce fluid. And the yolk sac develops as part of the blastocyst. It produces primitive red blood cells, and soon it's incorporated into the umbilical cord. The embryonic membrane begins to form at the time of implantation. The first and the outermost membrane to form is the chorion. This thick membrane develops from the trophoblast and has many finger-like projections, and these are called chorionic villi, and these are on the surface. Chorionic villi can be used for early genetic testing of the embryo at about 8 to 11 weeks. The second membrane to form is the amnion. The amnion is a thin protective membrane that contains amniotic fluid. The primary functions of the amniotic fluid are to act as a cushion to help protect the embryo against mechanical injury and to help control the embryo's temperature. This relies on the mother to release heat. Okay, talk a little bit about twins. Twins normally occur in about 33.2 per 1,000 live births in the US. They can either be fraternal twins or identical twins. Dizygotic twinning increases with maternal age up to about 35 years old. And then after 35, it decreases abruptly. Dizygotic means a male and a female twin. It could be from two separate ova fertilized by two separate spermatozoa. The current rate of twinning is attributed to the delayed childbearing of women nowadays and the use of artificial reproductive treatments. So dizotic from two separate ova that's fertilized by two separate sperm or spermatozoa. They have two placentas. They'll have two chorions and two amnions. Sometimes the placentas may look like they are one or joined together because they kind of fuse and they just appear to look like they're one. Fraternal twins, they're no more similar to each other than they would be if they were born individually or singly. They may be of the same or different sex as twins. This is a picture of fraternal twins developing. Again, two sperms, two ovas. You end up with two chorions, two amnion sacs, and usually two placentas. This is one, and here's one. Okay, identical twins, they develop from a single fertilized ovum, one egg. They're normally the same sex, they have to be the same sex, and they have the same genotype. Identical twins usually have common placenta, so they share a placenta. Their monozygosity is not affected by environment or race or physical characteristics or fertility issues. So identical twins are not affected by the environment, race, or any other issue like that. Both fetuses are the same sex and they will have the very same characteristics. Monozygotic twins originate again from the division of the fertilized ovum and at different stages of early development. The number of amnions and chorions present will depend on the timing of that division. So some identical twins are divided early on, and then some later on during the process, which we'll talk about here in just a second. Okay, so this is the different time frames. If the division happens within 4 days after fertilization, you're going to have two embryos, you'll have two amnions and two chorions. The amniotic sac will eventually be covered by a common chorion, or they'll share the common chorion, and the monochorionic placenta. They'll have a monochorionic placenta that will be shared. If division occurs about day 4 to day 8, you'll have two embryos, but they will already have a separate amnion sacs, and they will share a placenta. If an amnion already has developed, remember that fetal membrane, if it's already developed, division at 8 to 12 days after fertilization, we'll have the two separate amnions. Sorry, the one. So you'll have two embryos with a common amniotic sac and a common chorion, right? Because it's already developed before the division. So monochorionic and monoamniotic placenta. And then on rare occasions, identical twins are a little more rare than fraternal twins. So they only occur about 1% of the time. Monozygotic twins originate from division of the fertilized ovum again at different stages of early development, and that's after the zygote consists of thousands of cells. Usually the division will occur within about 4 days, and that's about 25% of the time. Monozygotic twinning is considered a random event, and it occurs in approximately 3 to 4 per 1,000 live births. Survival rate of monozygotic twins as a group is about 10% lower than that of dizygotic twins or fraternal twins. And congenital anomalies are more prevalent in these identical twins. Both twins may have the same malformation. All right, this is the picture 7-7B of the formation of identical twins. And you can see there's one ovum, one blastocyst, and then that, after it's fertilized, divides into 2, which is going to make them identical. Okay, umbilical cord. So the umbilical cord is being formed from the amnion as the placenta is developing. The body stalk fuses with the embryonic portion of the placenta, and this helps to provide circulatory pathways from the chorionic villi to the embryo. Vessels in the cord decrease to 1 large vein and 2 smaller arteries. So 1 large vein, 2 smaller arteries. A specialized mucoid connective tissue surrounds the blood vessels, and that's known as Wharton jelly. Central insertion into the placenta is usually considered normal, although it can be inserted in other areas. The umbilical cord has no sensory or motor innervation, so it can't feel anything, no nerves, so it doesn't hurt when the umbilical cord is cut. The average cord is about 2 centimeters or 0.8 inches thick or across, and about 55 centimeters or 22 inches long. This tissue plus the high blood volume that pulsates through the vessels prevent compression of the umbilical cord in utero. Umbilical cords appear to be twisted or spiraled, which is most likely caused by fetal movement. A true knot in the umbilical cord rarely occurs. If it does, the cord will be longer than usual. More common are so-called false knots, which are caused by the folding of cord vessels. A nuchal cord is said to exist when the umbilical cord encircles the fetal neck. There's about 1 in 200 umbilical cords have only 2 vessels, an artery and a vein. This condition may be associated with other congenital malformations, primarily of the renal, the gastrointestinal, and the cardiovascular systems. This is a picture of the inside of the cord, or the umbilical cord, and a picture of the long umbilical cord attached to the placenta. We'll talk about this in a second too. There's 2 sides to the placenta, the fetal side and then the maternal side, and they look different. Okay, placenta. The placental development begins the 3rd week of the embryonic development. It develops at the site where the embryo attaches to the uterine wall. The function of the placenta is metabolic and nutrient exchange between the embryonic and maternal circulations. Excuse me. Placenta has 2 parts, again, a maternal or fetal side. Things to remember, the placenta enlarges until about the 20th week of gestation, and this continues to supply nutrients and metabolic activity between the fetus and maternal circulation. Smoking constricts blood vessels and decreases circulation to the fetus. Smoking causes vasoconstriction that can interfere with placental circulation. The infant may suffer negative effects, which include intrauterine growth restriction, However, lung problems should be avoided during pregnancy. Things that cause lung issues or lung problems. The placenta maternal portion consists of the decidua basalis and its circulation. It's here on the left, and the surface appears to be red and flesh-like or meaty looking. That's the circulation to the maternal side. The fetal portion consists of the chorionic villi and their circulation. The surface is covered by the amnion. The surface appears shiny and gray, really slick. Let's see, the completion of the maternal and placental embryonic circulation occurs about 17 days after conception, when the embryonic heart begins functioning. By 14 weeks, the placenta is a discrete organ. It produces glycogen, cholesterol, and fatty acids, and it continuously produces these for fetal use and hormone production. Fetal blood flow flows through the two umbilical arteries to the capillaries of the villi, and oxygen-enriched blood flows back through the umbilical vein into the fetus. This is an embryo at 5 weeks. The embryo has a marked C-shaped body with a rudimentary tail right here. The stage of the embryo starts on day 15, which is the beginning of the 3rd week after conception, and it continues until approximately the 8th week. So that's the embryo. Tissue differentiates into essential organs, and the main external features develop. The embryo is most vulnerable to tetragens again during this phase. At 3 weeks, a single tubular heart forms just outside the body cavity of the embryo. At 5 weeks, the optic cups and the lens vessels of the eye form, and the nasal pits develop. By day 35, the arm and the leg bugs are well developed. The heart, circulatory system, and brain show the most advanced development during this time. The brain has differentiated into 5 areas, and 10 pairs of cranial nerves are now recognizable. At 4 weeks, the fetus is 4 to 6 millimeters. The brain is formed to the from the anterior neural tube. Limb buds are seen, and the heart is beating. GI systems are beginning. At 6 weeks, we have a primitive skeletal shape. We have chambers of the heart present, and respiratory system is beginning. 12 weeks, 8 centimeters in size. Ossification of skeletal bones begin. The liver produces red blood cells. The palate is complete in the mouth. The skin's becoming pink. You have a thyroid hormone present, and insulin is also present in the pancreas. 16 weeks, about 13.5 centimeters, begin to form teeth. Meconium, which is the first bowel movement, begins to collect in the intestines. The kidneys are taking their shape, and they are getting hair on their scalp. At 20 weeks, they're about 19 centimeters. Myelination of the spinal cord is beginning. They can suck and swallow. Lanugo covers the body. That's the short, little, thick, fine hairs all over the body. Vernix also begins to protect the body, and that's that white, thick, creamy substance, whitish yellow, I guess, that helps protect the body. Surfactant is being produced now for the lungs, and the brain also appears mature at this time, 20 weeks. This is the fetus at 20 weeks. It weighs about 435 to 465 grams, or about 15.2 to 16.3 ounces. It measures about 19 centimeters, or 7.5 inches. All right, 28 weeks. Nervous system begins regulation of some functions. This has eyebrows and eyelids, and the eyes are also open at this time. 36 weeks is also beginning to have at 36 weeks sole creases. That's the lines in the feet. At 40 weeks, they're about 40 centimeters in length. They have enough surfactant to be able to breathe. Vernix is now just in the skin folds instead of all over the body. They're about 3,000 3,600 grams, or 6 pounds, 10 ounces to 7 pounds and 15 ounces. The skin is smooth now. Hair on the head is about 1 inch long, and the little body parts are kind of more plump, has a little bit of fat on them now. The fetus is considered full term at 38 weeks and up to 40 weeks after conception. This chart is in your book, table 7.4. You guys can look at that. It's kind of what we just went over with a little more detail on both of these. Okay, I put this slide up so you guys could actually see what a real baby at these weeks look like. I feel like it's better to see it here than actually in the hospital, where you're more surprised at the way they may look. Remember, these early babies come out not quite just cute and adorable when they're really early. They've got basic features. The skin is very thin and also kind of transparent at sometimes. But take a look at these, and if you guys have questions or in class, just be sure and ask me. Okay, this is a 12-week fetus. You can see it's very small. It's about 2.13 inches, and it weighs about 2 ounces. 16-week fetus, 7.32 inches, and it's about 5.15 ounces. And you can compare that to the size of a mom or nurse's hand, or the thermometer is not a whole lot bigger than a thermometer. 20 weeks. Again, this baby, you can really see the transparency of the skin, which makes it look very dark. This baby is about 10.12 inches and about 11.68 ounces. This is a 24-week baby, 12.68 inches and 1.32 pounds. Again, so very small. You can hold it in one hand. 28 weeks, 14.8 inches and 2.2 pounds. This one is 32 weeks, 16.93 inches and 3.75 pounds. This one looks a little more like a term baby. Looks like it might have a little extra tissue on it, but not a whole lot. All right, these are some questions, and we're just going to go over those in class, but I hope you enjoyed this presentation. I'm just amazed at how all this kind of works, and be sure to take time to view all these very carefully. Just the process is a lot. I know it's a lot to